So good afternoon, everyone. Good afternoon, ma'am. So we are the Group 6B and we will present our case finding. So our case finding is entitled Microbiology Case Study of 38-year-old with intermittent straight catheterization. So next slide, please. So first is the case history. So, a 38-year-old man with history of secondary progressive multiple sclerosis complicated by neurogenic bladder requiring intermittent straight catheterization presented with fevers, chills, and weaknesses. So the clinical evaluation revealed hypotension and leukocytosis concerning for sepsis. So his urinalysis was positive for 3 plus blood, 1 plus leukocyte, esterase, and nitrites. So urine was sent to the laboratory for culture and grew bacteria with the below gram stain and colony morphology. So the colony morphology will be tackled in the laboratory findings. So next.
Good afternoon, everyone. I'm Redwan Lopez, and I will be discussing about the laboratory findings of our chosen microbiological case study. So here are the isolates which have been presented. First is the gram stain showing gram-negative bacilli, and the second picture is the sheep blood agar growing non-hemolytic gray-yellow mucoid bacterial colonies, as you can see. So next is the McConkey agar growing mucoid lactose fermenting or pink bacterial colonies. As you can see, they are seen as pink bacterial colonies. So based on the laboratory findings and bacterial colonies which appeared, there are two possible gram-negative species that contributed to the patient's infection. So the two species are Klebsiella and Rotella. So as you can see on the picture of the Klebsiella, it was somewhat the same to the gram stain as shown here. So as a medical technologist, you can conclude that Klebsiella is uh, the species that infected the patient. But still, there are series of tests conducted in order to confirm and identify what really caused the infection. So both are Klebsiella and Rotella are both gram-negative bacilli that form lactose fermenting yellow mucoid bacterial colonies, as you can see in the isolates. So next, the bacterial colonies appear mucoid because of the bacteria's polysaccharide capsule. Unlike with the gram-positive bacilli, the the cell wall of the bacteria has more content of the peptidoglycan. And unlike with the gram-negative bacilli, the high content is the polysaccharide capsule or polysaccharide. Distinguishing these two requires molecular analysis because their morphology are somewhat identical. So as you can see here, uh, the picture of the gram-negative bacilli, which are the Klebsiella and Rotella, have almost the same morphology. So the first process or procedure conducted was the urine culture. Under the urine culture, there are only one type of bacteria that was detected, and the case study was not yet been uh, identified here. So a mass spectrometry was conducted, and a Rotella species has been identified. Then, but still, it is not yet confirmed. So series of another tests were conducted, and that is the blood culture. So under the blood culture, there were two sets of culture. And here it was identified that it was contradicting to Rotella species, and that is the Klebsiella oxytoca. So the the case finder reviewed the urine culture. So a subtle second bacterial colony morphology was seen and confirmed as K. oxytoca. So here we can conclude that what infected the patient is really K. oxytoca. So for the conclusion and discussion, next reporters please.
Okay, conclusion, discussion. Next. Okay. Patient had a urinary conclusion infection, UTI, caused by Rotella and Klebsiella, which was the source of the patient's sepsis. Only the Klebsiella was detected in this bloodstream. And Rotella species are rare in human disease, but have been documented to cause bacteremia, UTI, conjunctivitis, and cholecystitis. Usually, the Klebsiella is found or usually pick up in the healthcare associated environments such as hospitals or in the ICU or nursing department. Usually, the common route of Klebsiella is the wound, the IV line sites, and catheters. In our case study, the patient had undergone catheterization, which was the source of the patient's infection. Once the Klebsiella go inside our body, it may develop into an infection. Kaya doon na nag-start. Usually, Klebsiella is found in our in the GI tract, in the mouth, and in the mouth or in our nose. Rotella species just rare in human. So next slide. K. oxytica and K. pneumoniae are the most frequently implicated species in human disease and may cause a wide spectrum of infections such as UTI, respiratory infections, enteritis and meningitis and bacteremia. So next.
So, so I'm Nathan Girandabio and I'm about to report about the patient's treatment. So, as recorded, antibiotic sensitivities that were susceptible to both type of bacteria is that the treatment used were ceftriaxone and then transition to cefpodoxime with the result of the patient's urosepsis. So next slide. So in general, Rotella susceptibility to microbial agents are not well studied and should be based on the antibiotic sensitivities of the strain. And in contrast with Klebsiella is well documented to have increasing antimicrobial resistance. And next. So both bacteria is resistant to ampicillin, carbenicillin, and ticarcillin, as well as expanded spectrum beta lactam drugs. And also, Klebsiella bacteria harbors plasmids with, which produce beta lactamase enzymes. So that is our report about the Klebsiella. So thank you.
Thank you. Thank you,
Thank
ma'am.
you. Thank you, ma'am.